Welcome, curious minds, to another deep dive. Have you ever caught yourself wondering why, after battling a really nasty bug, say chickenpox or maybe measles, you somehow seem to gain this almost magical resistance to it forever?
Yeah, or maybe you've noticed how some people seem to just, brush off every cough and sniffle going around.
Right. While others feel like they're constantly under the weather, like their body's defenses are just, permanently overwhelmed.
It's a common observation, definitely.
Well, today we are going to unlock the secrets behind one of the most incredible, intricate, and utterly vital systems protecting you every single second. Your immune system.
It's a fascinating topic, really fundamental to our survival.
We're embarking on a journey to really unpack the science of immunity, pulling back the curtain to reveal the hidden battles your body wages basically all the time.
From warding off common colds and seasonal allergies, things we all deal with, to mounting a really sophisticated defense against more serious, sometimes life-threatening invaders.
Your immune system is truly the unsung hero of your well-being. So our mission for this deep dive is to extract the most important nuggets of knowledge from our source material, the body's defenses, unpacking immunity and its mechanisms.
And we're not just here to tell you what immunity is, are we?
No, exactly. We want to illuminate how it works with such astonishing precision, why it matters so profoundly for your everyday health, and what its remarkable capabilities truly mean for you, the listener.
We'll go from ancient observations of devastating plagues, all the way to the cutting edge of modern scientific breakthroughs.
We'll dissect the two main types of immunity, delve into the ingenious mechanisms your body employs to protect itself.
And even touch upon the delicate balance, what happens when this remarkable system, doesn't quite work as it should, which can lead to significant health challenges, unfortunately.
It's going to be a truly fascinating exploration. So let's begin right at the very beginning. Where did the whole concept of immunity even originate? What does this powerful word truly mean?
Well, it's a word that's really steeped in history, actually. The term immunity traces its roots back to Latin.
Latin, okay.
Yeah, the word immunis, which quite literally translates to free of burden.
Free of burden, interesting.
Now, what's fascinating is that its original usage, it had nothing to do with biology or medicine at all.
Really? What was it then?
In ancient Rome, immunis was used to describe, for instance, Roman senators or certain privileged citizens who were exempt from specific taxes or legal prosecution.
Ah, okay. So they were free of the burden of taxes or legal trouble.
Exactly. They were protected from certain civic duties or legal entanglements.
So protection from legal obligation, not illness. How did that transition then? How did it jump to our physical health?
Well, over time, that powerful concept of being exempt, being protected or free from a burden, it just sort of gracefully transitioned into the biological and medical realm. Mm-hmm. It began to describe an individual who, having survived a particular disease, was now effectively free of the burden of contracting that specific illness again.
Okay, so surviving something meant you were free from getting it again.
And this wasn't just seen as a temporary shield. It really implied a lasting, often lifelong defense.
And in modern scientific terms, I assume the definition has broadened quite a bit since then.
Oh, absolutely. Today, when we talk about immunity, we're referring to the body's comprehensive and adaptive resistance to injury caused by microorganisms, their toxic products, or really any foreign substance that could cause harm.
It's a dynamic, layered defense system.
Exactly. The immune system itself isn't like a single organ you can point to, like the heart or the liver. It's an incredibly sophisticated distributed network,
right,
composed of countless specialized cells and just a vast array of valuable molecules like proteins. And these components don't work in isolation. They orchestrate this collective, highly coordinated response to what immunologists call foreign substances, or more precisely, antigens.
Okay, and an antigen, just so we're clear, isn't necessarily something inherently bad like a virus particle itself. It's more like a molecular signature.
That's a really crucial distinction.
Yeah.
An antigen could be a protein on the surface of a virus, or a specific sugar molecule on a bacterium, or even a chemical released by a parasite.
Anything foreign.
It's the thing that your immune system recognizes as not belonging to your own body, not me. Mm-hmm. And this recognition of foreign versus self is just fundamentally important, because the immune system broadly concerns itself with reacting against any foreign antigen.
So it goes beyond just infectious diseases.
Way beyond. It extends its protective reach to things like toxins, abnormal cells like cancer cells, or even environmental allergens. It's constantly performing this incredible discrimination. It's quite amazing.
It is mind-boggling, especially when you think that people observed these protective phenomena centuries, millennia ago, long before they had any concept of cells or molecules. Microscopes didn't even exist.
Right.
The source mentions Thucydides, the famous Greek historian, chronicling the devastating plague in Athens way back in 430 BC.
Indeed. Thucydides, in his meticulous account of that horrifying plague, he made a truly profound observation, really one that laid a foundational stone for understanding immunity.
What did he notice?
He noted with striking clarity that during the plague's peak, only those individuals who had already recovered from the disease could safely nurse the sick without succumbing to it themselves.
Yeah.
They seemed to possess this, inherent protection, a shield against getting the illness again. This was a foundational aha moment for humanity, a clear, if maybe intuitive, understanding that surviving an illness could confer lasting acquired resistance.
That's astonishing. Just pure observation leading to such a key insight. And it wasn't just in ancient Greece, was it? This idea of exposure leading to protection seems to have popped up elsewhere, too.
That's right. Our source mentions that as early as the 5th century AD in ancient China, there were these remarkable practices of smallpox inoculation.
Inoculation, like early vaccination.
Sort of a proto-vaccination technique,
Mm-hmm.
yeah. It involved exposing healthy individuals to material from smallpox lesions.
How did they do that? Sounds risky.
Oh, it's definitely risky. Typically by scratching dried scabs into the skin or, get this, blowing powdered scabs up the nose.
Yeah, okay.
And while it often resulted in a mild form of the disease, it demonstrated an intuitive grasp that a controlled, perhaps lesser exposure could lead to protection against the full, often fatal disease later on. It was a bold step long before the scientific principles were understood at all.
So from these early, almost accidental-seeming observations, we jump forward quite a bit to the late 19th century. This is when scientific pioneers really started to systematically chip away at the mystery of how this protection actually worked, didn't they?
Absolutely. That was an incredibly fertile period for medical science, a real explosion of discovery. In 1890, you had two brilliant minds, Emil von Behring and Shibasaburo Kitasato.
Right.
They conducted experimental work that provided the very first kind of mechanistic insight into immunity. They showed something truly revolutionary at the time.
What was that?
They demonstrated that there were serum elements, so components floating around in the liquid part of the blood, that could actually transfer protection against infections.
Transfer protection, how?
Imagine the surprise. They showed that if you took serum, the blood liquid from an animal that was already immunized against something, say tetanus or diphtheria, and you injected that serum into a susceptible animal, one that hadn't seen the disease, the recipient would gain protection.
Wow. So the protection was literally in the blood.
Exactly. Something soluble was carrying the immunity. This monumental discovery laid the conceptual groundwork for what we now understand as humoral immunity. That's immunity mediated primarily by antibodies circulating in the blood and other body fluids, the humors.
Humoral, okay. And what's particularly fascinating is that around the same time, another scientist was looking at immunity from a completely different angle, wasn't he? But he arrived at an equally crucial piece of the puzzle.
Precisely. It's like they were examining different facets of the same gem, concurrently, but working independently. Elie Metchnikoff, back in 1884.
Metchnikoff, right.
He was observing the cellular world under his microscope. He saw these spiralized white blood cells, which he famously termed phagocytes, literally meaning cellular eaters, actively ingesting and destroying microorganisms. Bacteria, things like that.
Eating them up.
Yep. But what truly grabbed his attention was that these phagocytes seemed significantly more active and more abundant in animals that had already been immunized.
Ah, so immunization boosted these cell eaters.
It seemed so. This was a critical revelation because it pointed directly to a powerful cellular contribution to immunity. It showed that not all defense was mediated by factors just floating in the blood. Cells themselves were active participants.
So you had Behring and Kitasato showing the soluble factors, the humoral side, and Metchnikoff showing the cellular side.
Exactly. And what's truly fascinating here is how these early, seemingly disconnected observations and experiments began to paint this increasingly detailed picture of a complex, multifaceted defense system.
Long before they had the tools we have now.
Right. Long before we could see the intricate molecular machinery, these pioneers identified that the body doesn't rely on just one strategy. It's not just soluble factors or just cells. It's this symphony of defense, using both humoral and cellular components, different angles of attack to ensure comprehensive protection.
It really highlights the power of just keen observation and clever experimentation.
It really does.
That's an incredible start to our understanding. So, if those foundational discoveries laid the groundwork, let's dive into the two main types of immunity scientists have since clearly defined. First up, the immunity you're essentially born with, it's called innate immunity. I like your analogy. It's like your body's built-in, always-on security system.
That analogy is spot on. Innate immunity is also often called natural immunity because, as you said, it's inherent from birth. It's deeply woven into our genetic and constitutional makeup.
So just there, ready to go.
Consider it your immediate, non-specific response system. It acts as the body's very first line of defense, ready to spring into action instantaneously upon encountering any perceived threat.
And a key thing is it doesn't get better with practice, right?
That's a defining characteristic. Its effectiveness is not improved or enhanced by prior exposure to pathogens. It doesn't learn or get better from previous encounters. And crucially, it lacks specificity or immunological memory. Meaning it doesn't really differentiate between, say, a measles virus and a common cold virus. It just recognizes this is foreign, initiate attack protocols. It treats all invaders similarly at this stage.
Like a general alarm system, not a precision weapon. And our source material even dives into some fascinating classifications of this innate immunity, showing how deep this built-in protection runs.
Absolutely. These classifications really highlight the pervasive and varied nature of innate immunity, showing its influence even within a species. First, there's individual immunity.
Okay.
This refers to the inherent variations in resistance or susceptibility to infection that we can see among different individuals, even within the same race and species. These differences are largely genetically determined.
So like why I might get a cold but you don't, even if we're exposed to the same virus.
Exactly. The source notes a striking example with tuberculosis in twins. If one identical homozygous twin develops TB, there's an exceptionally high probability the other twin will too, much higher than with non-identical heterozygous twins.
Wow.
This dramatically underscores how our specific genetic blueprint can predispose us to greater susceptibility or, conversely, greater inherent resistance to certain pathogens.
So some people genuinely are just naturally more resilient due to their unique genes, even in similar environments. That's quite something.
It is. Then we have racial immunity. This describes differences in susceptibility or resistance observed among different human races, or even among different breeds within a single animal species.
Examples.
A classic one cited is the remarkable resistance of Algerian sheep to anthrax, a severe bacterial infection they just seem to shrug off compared to other sheep breeds. In humans, a particularly powerful and well-studied example is the genetic resistance to Plasmodium falciparum malaria found in certain human populations. This is often linked to carrying the sickle cell trait.
Ah, the sickle cell connection.
Right. While full-blown sickle cell disease causes significant health problems, carrying just one copy of the sickle cell gene confers a substantial protective effect against severe malaria. It's a fascinating evolutionary trade-off, isn't it? Where a genetic variation that can be harmful in one context proves beneficial in another due to environmental pressures, like living where malaria is common.
That's a stark example of natural selection literally shaping our immunity. And finally, there's species immunity. That sounds like the broadest level.
It is. Species immunity refers to the total or relative resistance to a specific pathogen shown by all members of a particular species.
Like humans not getting Dutch elm disease.
Exactly. Or chickens being naturally resistant to Bacillus anthracis, the bacterium causing anthrax in other animals like cattle or sheep. And thankfully, we humans are completely unsusceptible to most plant pathogens, or many specific animal pathogens like rinderpest, which devastated cattle populations. We simply don't get sick from those specific diseases.
Why is that? What makes a whole species resistant?
It really comes down to the underlying physiological and biochemical differences between host species. Pathogens are often highly specialized. They need very specific conditions to thrive and replicate the right host body temperature, the precise receptors on host cells they need to latch onto, the specific nutrients they require, maybe even the host's specific cellular machinery or metabolic pathways.
So if a species doesn't offer that perfect environment,
then it's inherently resistant.
Yeah.
The pathogen simply can't establish an infection. It's a natural, fundamental barrier built into life itself, based on the compatibility or lack thereof between the microbe and the potential host.
That's incredible. Our very biology acts as a built-in species-specific barrier before anything even happens. So, now that we understand its scope, let's get into the ingenious mechanisms. How does our body actually do innate immunity? From the big stuff down to the tiny molecules.
It's truly a marvel of natural engineering, layer upon layer defense. The first line is often the simplest, yet incredibly effective. Anatomical or mechanical barriers. Think of your intact skin.
The outer wall.
Exactly. And also your mucous membranes lining your airways, your gut, etc. These are literally your body's physical walls, creating an impassable fortress, preventing most microorganisms from ever getting inside in the first place.
But they're not totally foolproof.
No, and the source wisely notes that any breach in this integrity, a small cut, an abrasion, even a needle stick injury, can unfortunately provide a direct route for infection. It's a vulnerability.
So even a tiny scratch can be a gateway.
It can. Beyond the skin, our internal surfaces are also meticulously protected. The mucous linings are constantly coated in this sticky layer of mucus.
Like flypaper for germs?
Kind of. It acts as a physical trap, ensnaring invaders. These linings also secrete acidic substances and potent enzymes, like lysozyme.
Lysozyme, what does that do?
It directly attacks and breaks down bacterial cell walls. It's like a chemical weapon against bacteria. And then a particularly elegant defense in the lower respiratory tract involves these tiny, hair-like projections called cilia.
Cilia, right. I remember those from biology.
They constantly beat in this coordinated wave, physically propelling mucus-entrapped microorganisms upwards and outwards, away from the lungs. It's like a sophisticated biological conveyor belt, constantly clearing debris.
Keeping the airways clean.
Yep. Furthermore, the constant washing action of bodily fluids like saliva, tears, and other mucus secretions physically sweeps away invaders from exposed surfaces. And these fluids aren't just water, they also contain their own arsenal of antibacterial and antiviral substances.
It truly sounds like a self-cleaning, fortified castle, constantly maintaining its defenses.
Okay, what about the physiological barriers? These sound more like the soluble sentinels, a chemical warfare happening within our body fluids.
Precisely. These are the chemical and biological defenses dissolved in our bodily fluids, often working invisibly. First, consider gastric acidity.
Stomach acid.
Right. Few ingested microorganisms can survive the extremely low pH shit, the intense acidity of our stomach contents. It acts as a highly effective natural chemical sterilizer for most things we swallow, dismantling pathogens before they can ever reach the intestines.
A harsh welcome.
Very. Then, as we mentioned, there's lysozyme. It's not just in mucus, but also found abundantly in tears, saliva, and other secretions. Its ingenious action is to directly break down peptidoglycans, which are crucial components of bacterial cell walls. It essentially dissolves and disarms many types of bacteria.
And interferon, that's a big one, especially with viruses, right?
Yeah.
Interferons, IFN, are absolutely crucial players, yes. Especially against viruses. These are a type of signaling protein, a cytokine, produced by cells primarily when they become infected with a virus.
What do they do? Kill the virus?
Not directly. Their primary function is remarkable. They act as an early warning system. They interfere with viral replication by signaling to neighboring uninfected cells. They basically tell those cells to batten down the hatches and prepare their defenses. They induce the production of antiviral proteins inside those neighboring cells, proteins that inhibit viral mRNA translation. Essentially, they prevent the virus from hijacking the cell's machinery to make more copies of itself.
So it's like a cellular alarm bell telling the neighborhood to lock down?
Pretty much. And interferons also play a broader role in initiating inflammatory reactions against bacterial pathogens too, so they're quite versatile molecular sentinels.
Okay. And the complement system. That sounds incredibly complex, almost like the secret weapon in the blood.
It is indeed incredibly complex and remarkably powerful. Think of the complement system as like a pre-primed string of biochemical dominoes, or maybe a tripwire defense system dissolved in our blood and tissue fluids.
Dominoes, I like that.
It's a complex cascade involving over 30 different components, various enzymes, regulatory proteins, receptors, all just waiting. Once the first domino falls, maybe triggered by recognizing a pattern on a microorganism's surface, a chain reaction is set off.
And what happens then?
This cascade rapidly amplifies the initial signal, culminating in really powerful outcomes. Either tagging the invader for cleanup by our phagocytes, making it tastier for them, a process called opsonization, or directly punching holes in the invader's membrane, causing it to burst and be destroyed. Direct lysis. It's an immediate, often explosive response, ready to go into action very quickly.
Wow, punching holes, that's pretty direct.
It is. And there are different ways to kickstart this cascade. The alternative pathway can be directly triggered by microbes early on, while the classical pathway usually needs antibodies, which come later.
Got it. And I saw something about defensins and catholicitins, too. Sounds like more weapons.
Yes, defensins and catholicitins are also very important. These are small but mighty antimicrobial peptides. Think of them as natural antibiotics produced by our own cells. They're found in various bodily secretions and within immune cells themselves.
But what do they do?
They mediate innate immunity by directly disrupting microbial membranes, basically poking holes in them like tiny molecular shivs, leading to the microbe's death. They provide a rapid, broad-spectrum defense, often acting as immediate, all-purpose molecular bodyguards before the more specific immune cells even fully mobilize.
Little molecular assassins.
Kind of. And alongside them are acute phase proteins. These are proteins whose concentration in the blood rapidly increases following tissue injury or infection. They help in various ways, including reducing inflammatory damage and assisting in directly fighting off the pathogen. C-reactive protein, or CRP, is a well-known example doctors often measure.
Okay, so we have the physical walls, the chemical warfare, and then we have the eaters, the phagocytes Metchnikoff saw.
Ah, yes, the cellular eaters. That brings us to phagocytosis. This is a foundational process. Specialized cells, primarily blood monocytes, which mature into tissue macrophages, and neutrophils literally engulf or ingest extracellular particulate material.
What kind of material?
This includes whole microorganisms, cellular debris from damaged tissues, dust particles, other foreign bits. They are the vigilant cleanup crew, and often the very first cellular responders arriving at the site of an invasion, constantly patrolling tissues and consuming threats.
And when you get a cut and it gets red, swollen, hot, and painful, that's the classic inflammatory response, isn't it? Part of this innate system.
Exactly. Inflammatory responses are a quintessential hallmark of innate immunity and are absolutely essential for both healing and defense. When there's local tissue injury or infection, the body mounts local inflammation.
Why? What's the point of the redness and swelling?
It's strategic. It directs immune components right to that injured or infected tissue. It happens through a coordinated series of events. Blood vessels in the area dilate, increasing blood supply that causes the redness and heat. Capillary walls become more permeable, leaky, allowing fluid and crucial immune cells, like neutrophils and monocytes, to exit the bloodstream and flood the affected tissue that causes the swelling and pain.
So it's flooding the zone with defenders and supplies.
Precisely. This influx of cells and fluid is vital for fighting the pathogen and initiating tissue repair. And in more severe or widespread infections, you might also see systemic responses, like a fever, or a general increase in the production of white blood cells, which are clear signs the body is ramping up its overall defense across the entire system.
It's a full-scale local mobilization, complete with reinforcements. And then there's a truly surprising ally in our defense mentioned in the source, our own resident bacteria. That seems counterintuitive.
It does, doesn't it? But it's a concept that's gaining increasing scientific recognition for its profound importance. Microbial antagonism. Our resident bacterial flora, the trillions of normal, mostly harmless bacteria thriving on our skin, and especially in our gut, they actually play a crucial, often overlooked role in innate immunity.
How? Aren't they just there?
They act as silent sentinels. How? Well, they literally compete with incoming pathogens for essential nutrients and physical space, often outcompeting the harmful microbes. They take up the parking spots, so to speak.
Right.
Furthermore, some of these friendly bacteria produce substances like bacteriocins that can directly inhibit the growth of pathogenic microbes, effectively preventing colonization by harmful invaders.
So they fight for us.
In a way, yes.
Oh.
It highlights the crucial importance of our normal flora microbiome and innate immunity. They're part of the team, a vast symbiotic network working to keep us safe and healthy. Maintaining a healthy microbiome is actually part of maintaining strong defenses.
That's a great twist, our own bugs helping out. And what about the NK cells? Natural killer cells. They sound pretty direct.
They are indeed. Natural killer NK cells are a unique type of lymphoid cell that mediates innate immunity. What makes them exceptional is their ability to directly recognize and destroy virus-infected cells and even some tumor cells without any prior sensitization.
Meaning they don't need to be trained first.
Exactly. They don't need to learn about the specific threat first, unlike the cells of the acquired immune system we'll talk about later. Their mechanism is clever. They recognize subtle changes on the surface of infected or cancerous cells. For instance, they look for cells that have suspiciously low levels of certain self-markers called MHC class I molecules, which are normally present on healthy cells.
Like checking ID badges.
Kind of. When they detect these missing self-signals or other stress signals, they swiftly trigger the destruction of those abnormal cells, eliminating threats on the spot before they can proliferate or spread. They're crucial for early control of viral infections and tumor surveillance.
And what's truly compelling here, as you mentioned earlier, is that it's not just about what you're genetically endowed with at birth, but also how well those built-in defenses are maintained throughout your life. It's holistic. Certain everyday factors can really influence how effective your innate immunity actually is, can't they?
That's a crucial insight, yes, and one that resonates deeply with our daily lives. While innate immunity is inherent, its efficiency and robustness can certainly be significantly influenced by several factors. For instance, age plays a profound role.
How so?
Individuals at the extremes of life, so fetuses, newborns, and the elderly, often show higher susceptibility to infectious diseases. This is because their immune systems are either immature and still developing, like in infants who haven't yet built a full repertoire of defenses,
Right.
or they're waning and becoming less robust, which happens in older adults. Immune cells might become less active, less numerous, or slower to respond. For example, the thymus gland, which is crucial for developing T cells, starts to shrink quite dramatically with age, impacting overall immune competence. This is called immunosenescence.
So it's not just chronological age, but the developmental stage or decline of the immune system itself.
Exactly. Another absolutely critical factor is nutritional status.
Diet matters.
Hugely. Malnutrition, particularly severe or chronic deficiencies in vitamins, minerals, or protein, can significantly impair both humoral and cellular components of immunity. To give you a tangible example, it can reduce the activity of neutrophils, those crucial phagocytic eater cells. It can diminish interferon responses, weakening that early viral warning system. And it can lower complement levels, reducing the potency of that biochemical cascade.
So if you're not getting enough essential nutrients,
Your body literally can't effectively mount its basic immediate defenses. It needs the right building blocks and ample energy to synthesize and maintain these incredibly complex and energy-intensive defense mechanisms. Your diet directly fuels or hinders your body's most fundamental defenses.
that's a powerful and very direct connection. Okay, what else?
And finally, hormonal levels also play a significant, if often overlooked, part. Disorders that disrupt normal hormonal balance, like poorly controlled diabetes mellitus or hypothyroidism, can increase susceptibility to infections.
How does diabetes affect immunity, for example?
Well, chronically high blood sugar levels in diabetes can impair the function of white blood cells, like neutrophils, making them less effective at engulfing and killing bacteria. High sugar also provides a more favorable environment for some microbes to grow.
It truly makes you realize how interconnected everything in our body is. The internal environment, down to our hormones and what we eat, plays this massive role in supporting or hindering these built-in defenses. It's not just a collection of parts.
Not at all. It's a beautifully holistic and integrated system.
Okay, so if innate immunity is the standing army always ready, broad attack, no memory, then acquired immunity is like the elite special forces. Highly trained, incredibly specific, and with an astonishing long-term memory. So what does this distinction truly mean when we talk about acquired or adaptive immunity?
That's an excellent way to put it. Acquired or adaptive immunity is the resistance that an individual develops and gains throughout their life. Unlike innate immunity, which is inherited, this system is a product of your body's encounters with different pathogens and foreign substances over time. It learns from experience.
And its key characteristics are much more sophisticated, right?
Extraordinarily so. It involves the specific recognition and elimination of pathogens, meaning it can tell the difference between measles virus A and flu virus B. It possesses immunological memory for rapid and enhanced future responses to things it's seen before. And it has the truly remarkable ability to discriminate with exquisite precision between self and non-self. It knows what to attack and, crucially, what to leave alone.
And who are the main players here?
This highly intelligent system relies primarily on two types of specialized white blood cells, the true masterminds. B lymphocytes, B cells, and T lymphocytes, T cells.
B cells and T cells. Okay. So this is the part of our immune system that actively learns, remembers, and adapts. And within acquired immunity, we have two major categories, active and passive. Let's dig into active immunity first.
Right. Active immunity is where your own immune system is stimulated to produce its specific defenses. It's a process induced by exposure to a foreign antigen, prompting your body to actively generate specific antibodies.
Those Y-shaped proteins.
Exactly, the ones that bind specifically to invaders. And also to generate activated T cells, which are crucial for directly killing infected cells or helping coordinate the overall response.
And it takes time to build up.
Yes, the development of active immunity typically occurs after a latent period. It takes some time, maybe days or weeks, for your body to fully build up this protection after the first exposure. But the significant advantage is that this protection is usually very long-lasting, sometimes for a lifetime.
And within active immunity, there's natural active immunity. This is the one we usually think of, right? Like in chickenpox as a kid.
Exactly. Natural active immunity is acquired through, well, natural means, either through clinical infections where you actually get sick and have symptoms, or sometimes through subclinical infections where you're infected but might not show obvious signs, yet your immune system still learns.
And this is often lifelong.
Often, yes, for many viral diseases, measles, mumps, rubella, chickenpox. Once your body encounters these pathogens and successfully clears them, it usually retains a strong, lasting memory. However, it's really important to note, this natural active immunity can be short-lived for other pathogens.
Like the flu.
Like influenza, exactly. This is because the flu virus undergoes significant antigenic variation. It constantly changes its surface proteins, its appearance, making it look new to your immune system each season, or even multiple times a season. So your previous immunity might not recognize the new version well.
So that's precisely why we need a new flu shot every year, because the virus keeps changing its disguise.
Precisely. And the source mentions an intriguing concept called premonition in some specific cases, like maybe syphilis or malaria in certain contexts.
Premonition, what's that?
Here, immunity seems to last only as long as the original infection persists, perhaps at a low level in the body. It provides protection against reinfection with a new strain, but only while the original pathogen is still present. If the original infection is completely cleared, that specific immunity might wane, leaving the individual susceptible again. It's a very specialized situation.
Interesting. So that's nature's way of teaching our immune system. What about the human-engineered version, artificial active immunity? This is vaccines, isn't it?
You're absolutely right. Artificial active immunity is skillfully induced by vaccines. These are ingenious preparations. They might be live but weakened, attenuated microorganisms or killed inactivated microorganisms or even just specific components or products derived from them.
And the goal is?
They are meticulously designed to safely mimic a natural infection, exposing your immune system to the key antigens without actually causing the full-blown disease. The core goal is to safely teach your immune system to recognize the threat and build strong, specific protection, including memory.
And there are quite a few different types of vaccines now, reflecting some incredible scientific ingenuity over the years.
Indeed, the field of vaccinology is incredibly diverse and constantly evolving. We have live attenuated vaccines. These contain live organisms that have been carefully weakened or attenuated in a lab, so they have significantly reduced virulence. They don't cause serious disease in healthy people.
Why use live ones?
Well, they're often highly effective because they can still multiply a little bit within the body, providing continuous antigenic stimulation. This typically leads to very durable and robust immunity, often mirroring the protection you'd get from a natural infection, sometimes after just 1 or 2 doses.
Examples.
Classic examples include the BCG vaccine for tuberculosis, the oral polio vaccine, and the measles, mumps, and rubella MMR vaccine, yellow fever vaccine too.
But using a live, even if weakened, organism, that sounds like it might have its own considerations, maybe for people with weakened immune systems.
That's a crucial point, yes. While highly effective, live attenuated vaccines are generally not recommended for individuals with severely compromised immune systems, say, from advanced HIV, certain cancers, or immunosuppressive therapy. There's a small theoretical risk they could cause disease in such vulnerable populations.
Okay, so what's the alternative?
Then we have killed inactivated vaccines. These contain whole organisms that have been rendered inactive or killed, usually by heat or chemicals, so they absolutely cannot replicate in the body.
Safer, maybe.
Generally considered safer from the perspective of causing infection, yes, as there's no risk of the pathogen reverting to virulence. They still stimulate active immunity. However, because they don't multiply, they often stimulate a less vigorous or long-lasting immune response compared to live vaccines.
So you might need more shots.
Exactly. They typically require multiple initial doses and often periodic booster shots to achieve and maintain sufficient protection. Many common vaccines fall into this category, like the inactivated polio vaccine, IPV, or the standard flu shot.
Okay, so sometimes a trade-off between absolute safety regarding infection risk and the robustness or duration of the immune response. What about something like tetanus shots? That's not the whole bacteria, is it?
Good question. No, it's not. That's where toxoids come into play. These are specifically modified toxins, the poisonous substances produced by certain bacteria like tetanus or diphtheria. The toxins have been treated usually with chemicals, so they retain their antigenicity, their ability to stimulate an immune response, but they completely lose their toxicity. They can't poison you.
So you become immune to the poison, not the bacteria itself.
Primarily, yes. They're used to protect against diseases where the main damage comes from the toxin produced by the pathogen rather than the pathogen invading tissues directly. Tetanus toxoid and diphtheria toxoid vaccines are excellent examples. They train your body to neutralize the poison.
Clever. And modern science has given us even more precise vaccines, right? Down to specific molecular parts.
Absolutely. The advances have been astounding. We now have subunit vaccines. These are revolutionary because they use only purified fragments of the major immunogenic components of a pathogen, essentially. Just the specific bits, usually proteins or sugars, that are most effective at triggering a protective immune response.
So not the whole bug, just the important parts.
Exactly. These are often produced using cutting-edge recombinant DNA technology, allowing for incredibly precise and safe vaccines. The hepatitis B subunit vaccine is a prime example, made from just a surface protein of the virus produced in yeast cells. Cellular pertussis vaccines are another example.
Wow. And then there are the really cutting-edge genetic vaccines, like mRNA vaccines.
Yes, those represent a major leap forward. Genetic vaccines harness our own cellular machinery. These use pathogen-specific genetic material, either RNA, like the recent COVID-19 vaccines, or DNA segments.
How do they work?
When administered, these genetic instructions are taken up by our own cells, which then temporarily produce the specific pathogen protein encoded by the vaccine's genetic material. Our immune system then sees this foreign protein produced by our own cells and mounts an immune response against it, leading to both humoral antibody and cellular T-cell immunity without ever exposing the individual to the actual pathogen.
It's like turning your own cells into a temporary safe antigen factory.
That's a great way to describe it. It's really quite remarkable. And the source also mentions anti-idiotype vaccines, though these are perhaps more theoretical or experimental still.
Anti-idiotype, that sounds incredibly specialized. What's the idea there?
It's a more conceptual, complex strategy. Essentially, you make antibodies against the antigen binding sites, the idiotypes, of other antibodies. So imagine you have an antibody that binds perfectly to a virus. An anti-idiotype antibody would bind to the binding site of that antibody. In doing so, the anti-idiotype antibody might mimic the shape of the original virus antigen.
Like a molecular mirror image.
Kind of. So this anti-idiotype antibody could then potentially be used as a vaccine itself, stimulating an immune response that targets the original virus. It's like using a mold of a key to make a new key. Highly specialized, still largely in research, but another ingenious theoretical approach.
Okay, that's mind-bending. With all these different types and strategies, what factors ultimately influence how well these vaccines work, or how strong and lasting an antibody response a person actually gets?
That's a critical question for both public health policy and individual protection. Many factors can influence antibody production and overall vaccine efficacy. There are intrinsic factors like your own genetic makeup. Some people just naturally mount stronger responses than others. And your overall nutritional status, as we discussed with innate immunity, is crucial here too. Your body needs the right fuel to build antibodies and power immune cells.
And external factors.
Yes. Things like the specific route and dose of antigen administration matter. Injecting into muscle might work better for some vaccines than taking them orally, for instance. Your age at the time of vaccination is important. Infants and the elderly sometimes have weaker responses. And the presence of any immunosuppressive agents you might be taking, perhaps due to other medical conditions or treatments, can dampen the response significantly. All these variables can impact the strength, speed, and duration of your immune response to a vaccine.
Makes sense. And then the ultimate genius, the very core of what makes active immunity so powerful and truly adaptive, the difference between a primary versus secondary immune response. This is really the heart of how our body remembers past illnesses and learns from them, isn't it?
This distinction is perhaps one of the most brilliant and elegant aspects of acquired immunity.
Does it?
Precisely why vaccines are so effective and why you typically only get diseases like measles once. When you encounter an antigen for the very first time, let's say your first exposure to the measles virus, or your first dose of a measles vaccine, your body mounts what we call a primary response.
Which is?
This initial response develops relatively slowly. It often takes days, maybe even a couple of weeks, to reach peak effectiveness. During this time, your immune system is essentially learning about this new invader. It's identifying its unique molecular signatures, activating the right B and T cells, getting them to proliferate, and starting to produce antibodies and effector T cells from scratch. It's like a reconnaissance mission, followed by setting up a brand new, highly specialized defense system for the first time.
So it's a slow burn, a foundational learning period. But then the magic happens with the next encounter, right?
Exactly. Upon subsequent encounters with the same antigen, maybe you're exposed to measles again years later, or you get a booster shot, your immune system doesn't start from zero. Instead, it launches a secondary response. And this is where the genius of memory truly shines.
How is it different?
This response is significantly faster, kicking in within hours or days, not weeks. It's exponentially stronger, producing much higher levels of antibodies and more effector cells. And it's much more sustained than the primary response.
Why? What makes it so much better?
It's mediated by those specialized, long-lived memory T B cells that were generated and selected during that initial primary exposure. These memory cells don't just remember, they're primed and ready for rapid action. They can quickly proliferate and differentiate into effector cells upon seeing the antigen again, overwhelming the invader often before it can establish a significant foothold or even cause any symptoms. It's the body's sophisticated learning curve, turning a slow initial identification into a rapid, powerful, and often silent counterattack.
That's a truly elegant solution to recurrent threats, a biological hard drive for invaders. Okay, so that's active immunity, your body doing the heavy lifting, learning, and building its own long-term defenses. But what if you need protection right now? Or you're maybe too young or too sick to build up your own immunity effectively? That's where the idea of borrowed protection or passive immunity comes in.
That's a perfect way to phrase it, borrowed protection. Passive immunity involves the direct transfer of ready-made antibodies from one individual who is already immune to another recipient, or sometimes from an immunized animal to a human.
And the benefit?
The significant and immediate benefit is that it provides instant protection. There's no latent period. The protective antibodies are immediately available in the recipient system to fight off the pathogen or neutralize a toxin.
But there's a catch, right?
Yes. The crucial drawback is that this protection is inherently transient or short-lived. This is because the recipient's own immune system wasn't stimulated to produce these antibodies. They didn't make them themselves. So once the transferred antibodies naturally degrade or are cleared from the system, which usually happens over weeks to months, the protection wanes and disappears. It's like borrowing a very effective shield for a dangerous moment rather than forging your own long-lasting armor.
And the most natural and beautiful example of this is from mother to child, isn't it?
Absolutely. That's natural passive immunity, and it's a cornerstone of early life protection. During pregnancy, a mother's maternal IgG antibodies, IgG, is the smallest and most abundant class of antibody, and it's uniquely able to cross the placenta or actively transported from her bloodstream to the developing fetus.
So the baby gets mom's antibodies before birth.
Exactly. This provides a crucial initial layer of defense for the newborn against pathogens the mother has previously encountered and developed immunity to. And then after birth, IgA antibodies, another type, are transferred through breast milk, particularly in the colostrum, the early milk.
What do the IgA antibodies do?
They offer additional localized protection, particularly coating the surfaces of the newborn's gastrointestinal and respiratory tracts, preventing pathogens from binding there. So this combined placental transfer and breastfeeding offers limited but vital temporary protection to the newborn during those vulnerable early months when the baby's own immune system is still immature and developing its own responses.
So breastfeeding isn't just about nutrition, it's literally providing a temporary immune shield too. Amazing. And then there's artificial passive immunity where medical intervention is used to provide this borrowed protection.
Yes. Artificial passive immunity involves deliberately administering preformed antibodies, typically in the form of antiserum, serum containing antibodies, often from humans or sometimes animals, or hyperimmune globulin, a more concentrated preparation of specific antibodies, directly to a person.
How would you use that?
It's often used for short-term, immediate protection, particularly in emergencies, or for highly dangerous exposures where there isn't time for the body to mount its own active response through vaccination, or if vaccination isn't possible or effective enough quickly.
Examples.
Common examples include giving rabies immune globulin after a bite from a potentially infected animal, tetanus immune globulin, TIG, after a potentially contaminated wound in someone not fully vaccinated, or hepatitis A or B immune globulin after a known exposure to those viruses. The immediate antibodies can neutralize the threat before the disease takes hold. It's also used to treat certain active infections like diphtheria or botulism by neutralizing the toxins.
But I imagine there are some downsides to just injecting antibodies from an external source.
There certainly are. Besides the obvious short lifespan of the transferred antibodies, they only last weeks to months, there's a potential for hypersensitivity reactions.
Like an allergic reaction.
Yes. Your body might recognize these foreign antibodies, especially if they come from an animal source like a horse, as foreign themselves, and mount an immune response against them. This can lead to allergic reactions ranging from mild to severe, or a condition sometimes called serum sickness, with fever, rash, joint pain. So while potentially life-saving in acute situations, it's generally a quick fix, not a long-term solution, and it comes with its own set of risks that need to be weighed.
So it's a powerful, immediate tool, but not permanent and not without risks. Is there ever a situation where you want to combine both? Get the immediate protection and start building your own long-term immunity?
Absolutely. That's where combined passive-active immunization comes in, and it's an incredibly clever and strategic approach used in specific circumstances. It's specifically used for situations requiring both immediate protection and the stimulation of the body's own long-term active immunity. The mechanism involves giving both things, usually at the same time but in different locations on the body. You give the preformed antibodies, like the immune globulin, for that crucial immediate protective cover. And you give the appropriate vaccine at the same time to stimulate the body's own immune system to start building its long-term active immunity.
So you get the best of both worlds.
Exactly. This dual approach is commonly employed for serious post-exposure prophylaxis, like after potential exposure to tetanus, rabies, or hepatitis B. The antibodies provide immediate defense, while the vaccine is busy training the immune system for lasting future protection. It's like having immediate bodyguard protection while also enrolling yourself in a comprehensive, personalized self-defense training program that will protect you later.
That's a fantastic metaphor for it. And all of this remarkable learning and adaptation is underpinned by that amazing concept we touched on earlier, immunological memory.
Yeah.
It really feels like the true magic of this acquired system.
Yes. Immunological memory is indeed the very core concept that allows acquired immunity to be so incredibly effective. And it's why we generally don't get certain diseases twice, or why vaccines provide long-lasting protection. After that initial exposure to an antigen, your immune system doesn't just forget. Instead, a small but powerful population of highly specialized, long-lived T and B memory cells are selected during the primary response, and they persist in your body for years, sometimes even decades.
They just hang around.
They do. These cells are essentially the immune system's historical record keepers and its rapid response units, primed for action. Their primary function is to enable that accelerated, heightened, and qualitatively superior secondary response upon any future encounters with the same antigen. They can quickly proliferate and differentiate back into powerful effector cells, plasma cells making antibodies, or killer T cells rapidly overwhelming the invader. It is the ultimate biological learning and recall system.
Ensuring that once the body has seen an invader, it's truly prepared for next time. Incredible. And finally, a truly crucial and delicate aspect of this whole sophisticated system is its ability to know who the enemy is versus who it's supposed to protect. The non-self-discrimination and tolerance. You said earlier this sounds like a tightrope walk.
It absolutely is a tightrope walk, constantly identifying threats while meticulously leaving healthy tissues untouched. This is a truly crucial ability of the immune system, to learn to avoid reacting against the body's own normal components, what we refer to as self. This profound process of recognizing and deliberately ignoring self is called self-tolerance.
How does it learn that?
It's a fundamental learning process that begins very early in development, mainly in the primary lymphoid organs like the thymus for T cells and the bone marrow for B cells. Immature immune cells that show a strong potential to react dangerously against the body's own tissues are either eliminated through a process called clonal deletion, or they're functionally inactivated, rendered anergic. The immune system is constantly being educated, almost like a highly refined security system being trained on what belongs and what doesn't.
So it weeds out the potential traitors early on.
Largely, yes, although there are peripheral tolerance mechanisms, too, that help keep things in check later in life. It's a complex, multi-layered system of checks and balances.
And what happens if that incredibly delicate learning process goes wrong, or if that self-tolerance breaks down later in life? What are the consequences?
If this self-tolerance breaks down, the consequences can be severe and debilitating. The immune system mistakenly attacks its own healthy tissues, leading to what we know as autoimmune diseases.
Autoimmune.
Yes. In these conditions, and there are many, over 80 known autoimmune diseases affecting various parts of the body, from joints and rheumatoid arthritis, to the thyroid in Hashimoto's or Graves' disease, to the nervous system in multiple sclerosis, or systemically in lupus. Your body's own powerful defenses turn inward, causing chronic inflammation and damage to its own organs and systems.
That's a tragic irony, isn't it? The system designed to protect you ends up harming you.
It is. It's a powerful testament to the incredible precision and self-regulation that the adaptive immune system normally maintains. This discrimination, this constant vigilance to protect self while eliminating non-self, is perhaps one of its greatest and most essential feats. When it fails, the results can be devastating.
Okay, we've explored the grand mechanisms, from the earliest observations right down to the intricate cellular and molecular details of how immunity works. But how does all this sophisticated machinery play out on a more, say, granular level, or even across an entire community? And as we just touched on, what happens when this remarkably intricate system falters? Let's start by zooming in on local immunity. What's that about?
Local immunity refers to the specific immune response that occurs right at the particular site where a pathogen initially invades or attempts to multiply. Think about the surfaces that are constantly exposed to the outside world. The mucous membranes lining your gut, your respiratory tract, your eyes, your mouth. These are the battlegrounds where the first skirmishes often happen.
So it's like defense right at the point of entry.
Exactly. And this on-site defense is primarily conferred by a very special type of antibody we mentioned briefly earlier, secretory SIGA.
Antibodies. Secretory IgA. So it's different from the IgG that crosses the placenta.
Yes, structurally and functionally. SIGA is the main antibody found in various bodily secretions like saliva, tears, breast milk, and especially in the mucus that coats the linings of the respiratory, gastrointestinal, and urogenital tracts.
And how does it work there? What's its mechanism?
Its mechanisms of action are ingeniously suited for these exposed surfaces. It primarily works by preventing microbes from adhering to the mucosal epithelial cells. It basically gums up their attachment sites, blocking their initial entry and stopping them from setting up shop. It also helps promote the physical removal of these microbes from surfaces, perhaps by clumping them together in the mucus to be swept away. And it can neutralize microbial toxins, enzymes, and even viruses before they can cause widespread damage or gain access deeper into the tissues or bloodstream.
So it's like the dedicated frontline defense, the border patrol, for those highly exposed mucosal areas.
That's a great way to think of it. Proactive interception right at the border.
And our source mentioned a really unique feature about SIGA, that it does not activate the complement system like other antibodies can. Why is that important for these particular locations?
That's a key distinction, and it speaks to the intelligent design, the fine-tuning of the immune system. Unlike IgG or IgM antibodies, SIGA generally does not activate that powerful complement cascade we discussed earlier.
Why is that a benefit here?
Because the complement system, while powerful for killing microbes, also induces significant inflammation as a byproduct. For delicate mucosal surfaces, like the lining of your gut, which needs to absorb nutrients, or your lungs, which need to exchange gas efficiently, constant or excessive inflammation would be highly damaging and disruptive to their normal function.
So SIGA provides protection without causing a big inflammatory mess.
Exactly. It provides quiet, non-destructive protection, helping to maintain the integrity and function of the mucosal barrier without triggering an inflammatory response that might harm the host tissues in these sensitive areas. It's a perfect example of the immune system tailoring its response for specific environments.
That's fascinating, a really nuanced approach for different parts of the body. Okay, now moving from the individual level to the collective, let's talk about herd immunity. This is a concept we hear a lot about in public health discussions, especially around vaccination. It seems to transcend just individual protection.
Herd immunity, or community immunity, is indeed a powerful concept. It describes the overall level of resistance to a particular infectious disease within an entire community or population group, rather than just focusing on whether one individual is immune or not.
And how is it achieved?
A high level of herd immunity is most often and most safely achieved through widespread vaccination, which builds up individual active immunity on a massive scale across the population. It can also contribute after widespread natural infection, but obviously with much higher risks of disease and death.
And how does that collective immunity benefit those who aren't immune? Maybe infants too young to be vaccinated or people with medical conditions preventing vaccination?
It creates this critical protective buffer, a collective shield. When a sufficiently high proportion of people in a community are immune, meaning they've either been effectively vaccinated or have recovered from the disease and have lasting immunity, the pathogen struggles significantly to find susceptible hosts to infect.
It can't find anyone to jump to.
Exactly. This dramatically slows or can even completely arrest disease transmission within the community. It effectively provides a human barrier that insulates those who aren't immune themselves. The virus or bacteria hits dead ends when it tries to spread. This concept is absolutely crucial for public health initiatives.
And it's been key to controlling diseases.
Absolutely fundamental. It's been key to the eradication of smallpox globally and the near eradication of polio in most parts of the world. It's the foundation for controlling measles, rubella, and many other vaccine-preventable diseases.
So if enough people are vaccinated, it essentially starves the pathogen of new hosts, and it can't spread, even protecting the most vulnerable among us.
Exactly. Conversely, epidemics are far more likely and can spread with alarming speed when herd immunity is low. If vaccination rates drop below a certain threshold, which varies depending on how contagious the disease is, there's a large pool of susceptible individuals created in the community, providing ample opportunities for the pathogen to spread unchecked, leading to outbreaks.
So herd immunity is really a powerful example of how individual health choices, like getting vaccinated, can have a direct measurable impact on the entire community's well-being.
It really is. It's not just about your individual protection. It's about contributing to the safety and health of everyone around you, especially the most vulnerable members of society, babies, the elderly, the immunocompromised. It underscores our interconnectedness in a very tangible way.
That really drives home the community aspect of health and the shared responsibility. But, okay, even with all these amazing, intricate mechanisms we've discussed, innate, adaptive, local, herd the immune system, like any complex biological system, can have its vulnerabilities. It can fail. What happens when this remarkably intricate system misfires or doesn't quite work as it should?
Unfortunately, despite its brilliance, it can. We generally categorize these as disorders of immunity, and they often fall into two main, often devastating categories. One major category is immunodeficiency.
Meaning too little immunity.
Exactly, where there's simply too little immunity, a weakened or deficient defense system. Deficiencies in immune function lead to a significantly increased susceptibility to infections, often recurrent, severe infections, sometimes caused by common microbes that wouldn't bother a healthy immune system. And often also an increased risk of certain malignancies or cancers, because the immune system also plays a role in spotting and eliminating abnormal cells.
So a compromised defense system leaves you vulnerable on multiple fronts. Are these always genetic, something someone is born with?
Not necessarily. Immunodeficiencies can be categorized as primary immunodeficiency. These are indeed genetic or developmental defects in the immune system itself. There are hundreds of different types known now affecting different parts of the immune system. These are typically present from birth or early childhood, meaning someone is born with a fundamental flaw in their immune machinery, perhaps lacking a crucial enzyme or a specific type of immune cell.
And the other type.
They can also be secondary immunodeficiency or acquired immunodeficiency. These develop later in life due to other diseases or external factors that suppress or damage the immune system. The most well-known example is probably HIV/AIDS, where the human immunodeficiency virus specifically targets and destroys crucial immune cells, called T helper cells, leading to a profound collapse of adaptive immunity over time. Certain cancers, particularly those affecting the blood cells or lymphoid system like leukemia or lymphoma, can also cause immunodeficiency. And importantly, immunosuppressive drugs medications often used after organ transplants to prevent rejection or to treat autoimmune diseases intentionally suppress the immune system, leaving individuals more vulnerable to infections as a side effect. Even severe malnutrition, as we discussed, can significantly impair immune function, leading to an acquired immunodeficiency state.
It sounds like a constant, exhausting battle just to stay healthy when your defenses are down. But then there's the opposite problem you mentioned, excessive immunity. Too much of a good thing. When the system becomes overzealous.
Exactly. In these cases, the immune response becomes overactive or misdirected. Instead of just protecting, it ends up causing collateral damage to normal, healthy tissues. This overenthusiasm manifests in a couple of key ways. First, we have hypersensitivity diseases.
Like allergies.
Precisely. Hypersensitivity is what we commonly refer to as allergies. These are exaggerated and often inappropriate immune reactions to otherwise harmless environmental substances, things like pollen, dust mites, pet dander, certain foods such as peanuts or shellfish, insect stings, or even some medications. Your immune system mistakenly perceives these benign substances as dangerous threats and launches a disproportionate, often uncomfortable, and sometimes even dangerous attack involving cells like mast cells releasing histamine and other inflammatory mediators.
So your immune system is basically misinterpreting a harmless signal as an invasion and massively overreacting, and the other manifestation of excessive immunity.
The other major category is autoimmunity, which we touched on earlier regarding self-tolerance. This is where the immune response mistakenly attacks and damages the body's own normal, healthy self-tissues. It's the ultimate tragic failure of self-tolerance. The immune system loses its critical ability to discriminate between self and non-self and turns its powerful weaponry inward.
Leading to diseases like rheumatoid arthritis or lupus.
Exactly. In conditions like rheumatoid arthritis, the immune system attacks the lining of joints. In type 1 diabetes, it destroys the insulin-producing cells in the pancreas. In multiple sclerosis, it damages the protective myelin sheath around nerve fibers. In lupus, it can attack multiple organs throughout the body. The immune system identifies components of these tissues as foreign, leading to chronic inflammation and progressive tissue destruction. It's an incredibly complex area of immunology, and understanding exactly why this tolerance breaks down in different individuals is a huge focus of ongoing research, aiming to develop better, more targeted treatments.
It's such a powerful reminder that balance is absolutely key, isn't it? Even within our own bodies. The immune system is a truly continuous marvel, constantly defending us, adapting, learning. But like any incredibly complex system, it can have its vulnerabilities, from underperformance in immunodeficiency to destructive overzealousness and hypersensitivity and autoimmunity. It really makes you pause and consider the silent, intricate warfare happening within us every single moment. Wow, we've journeyed through the incredible, often invisible world of immunity today. From its ancient origins and those remarkable early observations, through its intricate cellular and molecular mechanisms, innate and adaptive, right up to its profound impact on individuals and entire communities with concepts like herd immunity. You, our listener, now possess a far deeper understanding, I hope, of the incredible layers of defense your body deploys every single day, mostly without you even having to consciously think about it.
And remember, whether it's those inherited defenses you're born with, the innate barriers and the immediate responders like phagocytes and NK cells, or the learned adaptive responses involving B cells, T cells, and that amazing immunological memory that protect you throughout life.
Yeah.
Your immune system is truly a testament to the body's remarkable and dynamic capacity for protection, adaptation, and self-regulation.
Yeah, it really is a continuous, sophisticated battle fought tirelessly on your behalf, allowing us to thrive in a world that's actually full of microscopic threats.
Absolutely.
So thinking back on our deep dive, what stands out most to you? Perhaps it's the sheer biochemical ballet of the complement system, that incredible cascade of proteins working together in perfect synchronicity.
Or maybe it's the astonishing genius of immunological memory, allowing your body to remember invaders from decades ago and respond with such incredible speed and precision. That always amazes me.
Right. Or perhaps it's the surprising and crucial role of our friendly gut bacteria, acting as silent sentinels and key partners in fighting off pathogens who knew our own microbes were such allies.
It definitely adds another layer of complexity and wonder.
As we wrap up, here's a thought to ponder. With all these existing layers of defense we've explored, and given the constant evolution of pathogens that are always challenging our bodies in new ways, what new frontiers do you think immunology will explore next? What's the next big challenge or breakthrough needed to keep us truly free of burden from disease in the future?
That's a great question to leave people with. The field is certainly not standing still.
Definitely not. Thank you so much for joining us on this deep dive into the fascinating world of immunity. Until next time, stay curious and keep exploring.